If you've ever seen an action movie, you've probably seen that montage sequence where, the hero goes from being a total idiot to completely skilled in what they're doing. And that's what this lesson is going to be about. It's kind of like Mulan, but with B cells. We're going to talk about B cell maturation. And in this sketch, we'll follow a band of new recruits as they learn the ancient art of archery and strive to become the greatest archers in all of the immuno realm. But before we get started, let me explain why B cell development is so crucial. Basically, it accomplishes three main things. First, it produces a diverse array of B cells that are capable of recognizing a wide range of antigens. Second, it eliminates any defective or self-reactive B cells. And third, it allows B cells to travel to secondary lymphoid organs, like the spleen or lymph nodes, where they'll become mature, hopefully. So, since B cells develop in the bone marrow, and again, that's how they got their name, that's where we'll focus this sketch. And in fact, let's actually surround our training area with this giant bone fence to remind you that this whole scene is taking place inside the bone, in the marrow. Starting from the top, we'll introduce our king, Matt the Poet, and he's just there to remind you that B cells arise from hematopoietic stem cells. Those hematopoietic stem cells become common lymphoid progenitor cells before eventually transforming into either B cells, T cells, or natural killer cells. If you've seen our immune lineages sketch, you might remember that this is the right side, the lymphulets side. And so to emphasize this intermediary step, let's add this lymphulets in training banner prominently displayed beneath His Majesty. Now, to become a full-fledged B cell, a common lymphoid progenitor cell must go through 6 steps of development. And so as we go through these 6 stages, we'll work our way from left to right. So oddly enough, the very first thing a B cell has to do is become a pro. A pro B cell is the very first stage of B cell development. That pro stage is actually divided into 2. First, it's an early pro B cell, and to represent this, we have an early riser. He's working out with these heavy battle chains because the main thing that happens during the early pro B cell stage is heavy chain rearrangement. And let's take a step back for a quick second to review what's going on at the molecular level here. Every B cell is equipped with a B cell receptor on its surface, which is essentially just like an antibody. The B cell receptor has 2 distinct chains, a heavy chain and a light chain. The heavy chain contains V, D, and J segments, while the light chain contains V and J segments. There are many variations of these segments, and they can also undergo rearrangement, which means the segments get sort of cut and pasted together, which ultimately results in the creation of a totally unique chain. And that's what allows the B cell receptor to bind to such a wide array of antigens. So getting back to our early pro B cell, it's the D and J segments that get rearranged first. And that's why we've got this DJ. No good montage sequence is complete without a heart-pumping track behind it. And so we've got DJ, let's call him Joust-a-Lot, spinning or scratching these stone records to inspire our pros in training. And if you think about it, scratching those stone records is probably pretty hard. And so our DJ is getting a little sweaty, and he's wiping his face with a rag. That's a reminder that there are 2 special enzymes that splice D and J segments together, and those are RAG1 and RAG2. And just as a heads up, both maternal and paternal chromosomes undergo rearrangement, but once a chromosome has completed this event, the other chromosome is immediately just turned off. This process is called allelic exclusion, and it ensures that each B cell produces only one type of heavy chain. All right, so once the D and J segments come together, our cell is now a late pro B cell. During this step, the V segment is joined to the DJ segment. And that's represented here as a latecomer flashing a peace sign, which looks like a V, at the DJ. He's getting out of here because he's late for something. And there's actually one more step before we get to move on to our next stage of B cell development, and that's we need to recombine the mu gene with the heavy chains. The mu gene encodes the IgM constant region, which permits the heavy chain to actually attach to the B cell membrane. All right, so the first 2 steps are now done, and our late pro B cell can become a pre-B cell, and specifically a large pre-B cell. These names can get pretty annoying, and so to help you remember this order, we're going to move out of our pro workout area into our fitting preview space. This is where the new recruits get to check themselves out in their new armor and make sure everything fits just right. It's kind of like your white coat ceremony. Anyway, notice that we've also changed the color of the ground here. Now we're on grass. And that's just really to re-emphasize that we've moved on to a whole new part of B cell development. Now, we've got this giant archer here to remind you that we're talking about large pre-B cells. And he's trying on some light chainmail because the main event in this large pre-B cell stage is adding a surrogate light chain to the heavy chain to produce a pre-B cell receptor. Now, you'll notice that armor says loner on it, and that's because this is just a surrogate light chain. It's not the final thing, but we're just sort of test fitting everything here. The idea is to make sure the heavy chain can actually bind to the light chain and that it can be expressed on the surface of the B cell. So the heavy chain and the surrogate light chain are sent to the cell surface by a vesicle and placed near the side chains Ig alpha and Ig beta, which then signal to the B cell that it can go ahead and proliferate. The large pre-B cell then rapidly proliferates to produce small pre-B cells. That's sort of backwards from how you might expect it to go, but such is biology. The important point here is that the kappa and lambda light chains of the small pre-B cells, namely the V and J segments, undergo rearrangement, sort of like how this short stature recruit is sorting through or rearranging all these different colorful light chainmail armors. Okay, that's actually nothing like how it goes, but it is a good visual reminder. And remember that the kappa chains get rearranged first, followed by lambda chains. K comes before L. And we're moving on. Once the light chains have successfully rearranged, the small pre-B cell becomes an immature B cell. And how better to represent an immature B cell than this guy who is, well, behaving in the most immature way we could think of that you could also potentially watch in a public setting. It's possible we're pushing it a little on that one. Anyway, notice that he's wearing colorful light chainmail. That's a reminder that the light chains should already be rearranged by now. All right, so at the end of the montage sequence, there's always that one final test that the hero has to pass, and we've made it there. For B cells, that test is called clonal selection, and they have to pass it before leaving the bone marrow. Clonal selection includes positive and negative selection, and during positive selection, which we'll represent here with this positive target, B cells that have a strong affinity to MHC molecules are allowed to proliferate, while those that bind weakly or not at all die by apoptosis. Essentially, does this B cell work? Does it do its job and bind its targets? Then there's negative selection, or central tolerance. This is making sure that B cells don't bind to self-antigen. Self-reactive B cells, or those that do bind to self-antigen, are removed. You can remember negative selection with this no friendly fire sign. We don't want B cells that will attack our own tissue. If B cells fail either positive or negative selection, they undergo apoptosis, or programmed cell death. Basically, those recruits are thrown in the pit of death, and they're the ones who don't get movies made about them. All right, moving on, and I promise we're almost there. The surviving immature B cells exit the bone marrow, finally. From there, they migrate to secondary lymphoid organs like lymph nodes and the spleen, and we've got those sentinel towers out there in the background to remind you of that journey to lymph nodes. But they still have more names, I'm sorry to say, and so these migrating immature B cells are now called transitional B cells because they're still not quite yet mature. Some of these transitional B cells will go on to become follicular B cells. Those are the B cells that reside in the follicles of the germinal centers, hence this very hairy follicular archer. Other B cells differentiate into marginal zone B cells. Those are B cells that serve as the first line of defense against bloodborne antigens that have become trapped in the spleen. And so to illustrate these marginal zone B cells, we'll have this archer here sporting a spleen shield, and we'll stash them off to the side near the margins of the archway. And just like that, we've created some mature, naive B cells. They're called naive because they haven't been exposed to antigen yet. And when they do actually get exposed to antigen, they'll become activated. But we'll save those details for another sketch. All right, that was a lot to cover, so let's do a quick summary. First of all, B cells arise in the bone marrow from hematopoietic stem cells, which then become common lymphoid progenitor cells before eventually transforming, ultimately, into mature B cells. That process requires 6 main steps divided into 3 sort of stages, which we've illustrated with the ground under each stage. The 6 steps are as follows. First, an early pro-B cell is created, and this is when the D and J segments of the heavy chain rearrange. Next, in the late pro-B cell, the V segment is attached to the D and J segment. Then, the mu chain gets added to the heavy chain, and in the large pre-B cell, a surrogate light chain combines with the heavy chain to form the pre-B cell receptor. Signals are then sent to the large pre-B cell to proliferate into small pre-B cells. The kappa and lambda light chains of these small pre-B cells will then rearrange, and then these now immature B cells go through selection to remove any defective or self-reactive cells. The immature B cells then leave the bone marrow and travel to secondary lymphoid organs, where they can become mature naive cells, which include either follicular or marginal zone B cells. And finally, we've come to the end of this sketch. As you can see, it's not easy becoming an archer, but at least this DJ keeps things grooving. Good God, who writes this stuff?